Hello, today we will be reviewing Chapter 7 of our AP Biology textbooks, starting with the structure of cell membranes. The cell membrane is a selectively permeable membrane that regulates what enters and leaves the cell. The person who is credited with discovering the structure of the cell membrane is S.J. Singer. His model was named the fluid mosaic model because there are so many components of the cell membrane working together and moving as one. The cell membrane is made up of a double layer of phospholipids. Each phospholipid has a hydrophilic head and a hydrophobic tail. The cell membrane only allows lipid-soluble substances to pass through it. Think of toast as your cell membrane and butter as your lipid-soluble substance. The butter is able to melt into the toast because the toast allows it to. However, if the substance is not lipid-soluble, such as a brick in this case, the cell membrane will not allow it to pass through.
Transport is the movement of substances into and out of a cell. There are two types of transport, including passive and active transport. Active transport requires energy.
While passive transport does not require energy. She's really passed out.
Passive transport is movement down a concentration gradient from high concentration to low concentration, and no energy is required. Two examples of passive transport are osmosis and diffusion. Now let's go into more detail about those. There are two types of diffusion, simple and facilitated. But what's the difference between the two? Well, simple diffusion does not involve any protein channels, while facilitated diffusion does. An example of simple diffusion is the uptake of oxygen by a cell performing cellular respiration. The other part of passive transport is osmosis, which is the diffusion of water across a semi-permeable membrane.
Now we're going to talk about active transport. Active transport is the movement of molecules against a gradient, which normally requires energy, usually in the form of ATP. Pumps and carriers carry particles across the membrane by active transport. Active transport is responsible for the fact that cells contain a relatively high concentration of potassium ions, but low concentrations of sodium ions. The mechanism responsible for this is the sodium-potassium pump, which moves these two ions in opposite directions across the cell membrane. The electron transport chain in mitochondria consists of proteins that pump protons across the cristae membrane. The contractile vacuole in freshwater propista pumps out excess water that has diffused inward because the cell lives in a hypotonic environment. Exocytosis in nerve cells occurs as vessels release neurotransmitters into a synapse. Endocytosis is the process in which cells take in materials from the outside by engulfing and fusing them with its plasma membrane. Penocytosis is the uptake of dissolved particles. The plasma membrane invaginates around the particles and encloses them in a vesicle. Phagocytosis is the engulfing of large particles or small cells by pseudopods. The cell membrane wraps around the particle and encloses it into a vacuole. This is the way human white blood cells engulf bacteria and also the way in which amoeba gain nitrogen.
Now we're going to talk about hypotonic solutions, isotonic solutions, and hypertonic solutions. In a hypotonic solution, there is a lower solute concentration in the cell. That means that there is more water outside of the cell and it's going to move inside the cell. Think of low, hypo. In an isotonic solution, the solute concentrations are equal. This means that the percentage of water in the cell and the percentage of sucrose in the cell are equal. Think of water as an original movie and sucrose as the second movie. Usually somebody would want the second movie to be just as good as the first, like an equal sequel or squeakquel in this case. In a hypertonic solution, there's more water inside the cell, which means that water is going to move outside of the cell and there is a higher solute concentration. Think of how a windshield wiper moves water out, like a hyper wiper. For example, take the potato lab. The potatoes that were put in a hypertonic solution became plasmolyzed, and the potatoes that were put in a hypotonic solution became turgid.
And that's it for today's lesson on Chapter 7. Today we learned about cell membranes, passive and active transport, diffusion and osmosis, and hypertonic, hypotonic, and isotonic solutions.
What is that? What does that do? What does that mean? Is that so? What is the function of that? Osmosis Jones.
Wait.
Oh my.